Good evening, everyone. Thank you. My name's Alison Venn, and as Director of the Menzies Institute for Medical Research, it's really my pleasure to welcome you here this evening to the 2016 Menzies debate. To begin, the University of Tasmania wishes to acknowledge the Muhanina people, the traditional owners and custodians of the land upon which this campus was built. Many of you will be familiar with the work we do at Menzies, but there may be some of you for whom this is the first time you've come to one of our events, and I particularly want to extend a warm welcome to you. Menzies has been around for about 30 years now, and from small beginnings, we now have almost 300 staff and postgraduate research students working with us. As our mission states, we are here to perform internationally significant medical research that will lead to healthier, longer, and better lives for Tasmanians. And rather than explain in detail what we do, I'd like to play you a short video that we've had produced this year to support our philanthropic activities. And if I can find the little play bit. Where is that? Oh, yeah. Good.
If you or a loved one or someone close to you has had their life affected by multiple sclerosis, could you please stand? If you, a loved one, or a friend has been affected by dementia, could you please stand? If arthritis has affected you, someone in your family, or someone you know, could you please stand? If heart disease has affected your life, or the life of someone you love, could you please stand? Anyone whose life, or the life of a loved one, or the life of a friend who has been affected by cancer, please stand.
Thank you. I think that says it very well. Many of the people featured in that video were our volunteers at Menzies, and the main scenes were not scripted. People are genuinely standing in response to those questions, and I'm sure we would all be as well. The diseases that are mentioned there make up most of what we do at Menzies, but not all. And we like to use these opportunities for our debates to feature the kind of research that we have an interest in. And today we will be debating the stem cell revolution, and we'll be debating whether it's more hype than hope. So what are stem cells? I think we can think of stem cells as cells that haven't really decided what they want to be when they grow up. They are unspecialized cells that can renew themselves and make more stem cells, or they can decide to specialize and become another type of cell, such as a red blood cell, or a muscle cell, or a brain cell. Stem cells have been controversial at times. Some of that controversy comes from the use of embryos as a source of stem cells, including excess human embryos created by IVF and donated to research. Other options to generate stem cells are now available, but new controversies have emerged, and you'll hear more about them tonight. We chose this as our debate topic because it's an area of medical research that's of great public interest, but also of great confusion. And it's important to one of our research themes at Menzies, which is the neurodegenerative diseases and brain injury theme. And before we get into the debate proper, I need to do a couple of housekeeping things, let you know where the toilets are, which is over across the lobby, down the orange corridor and on the left. The fire exits are here just on my right, your left, and there's also a fire exit out the glass door just outside the main door at the top of the theatre. Sound quality, if you have trouble hearing, please let us know. So it's now my pleasure to introduce Bernie Hobbs, who is back here to moderate her third Menzies debate. Bernie has always had a passion for science. She's spent some time actually doing it, but her real passion is for talking about science. She's a former teacher and medical researcher. Bernie's been talking, writing, and apparently having a beer or two over science at the ABC since 1997. Whether it's through her radio show, award-winning websites, Science in the Pub, or the Experimentals, her mission is to convert the science equals yawn crowd. So thanks very much, Bernie. It's great to have you here again for our 2016 debate.
Thanks so much, Alison. Thank you. And I can guarantee it's not a hard sell getting me down here, because every year I think this event is probably one of, if not the most fun, and certainly the most publicly engaging event that I'm involved in each year. Last year we even had the honour of overturning the outcome of the debate by audience vote. So direct democracy, people, you're ruling the show tonight. Thanks so much for inviting me, and I will just quickly introduce our panellists this evening. Now, who's been to one of the debates before? See, it's so much fun, we just keep coming. Oh, joy, you've done your homework. Even some of the panellists have come in advance to check out how things work here. The debate's great because it really lets experts have a say on an issue without giving a lecture, without showing more than 3 slides, people. I'll be keeping and we've had some sneaks try and count an animation as one slide, but we know how many frames there are per second in those suckers, so we'll be doing a quick check on those as well. We're going to be very hard on the rules, but the main thing is our experts are going to be talking to us, covering some of the big issues in a really engaging and, where appropriate, light-hearted kind of way. And better still, not only do you get to judge before and after having heard from our contestants here tonight, you also get 20 minutes to ply them with any questions that you've got about stem cells down the track a little bit. So stem cells have been a word or a couple of words that we've heard of for more than a couple of decades now, and they've had a lot of promise. I mean, people like me who think as long as we've got our brain and our knees, old age shouldn't be too scary, but I'm only 51 and a half, so what the hell would I know? But I have also been, that talk about the knee stuff has been going on for a while. I'm not sure that we've got any evidence yet that the stem cell therapy there works, but we'll be hearing about that kind of stuff tonight because we're looking at hype versus hope. Now, you'll have no trouble guessing which side is the hype side tonight because you'll see the telltale indicator, a bow tie, featuring on our middle speaker on the hype team. So the hype team is clearly and firmly of the opinion that, and it's very important, I only have this one job, it's very important I get it right, that the stem cell revolution is more hype than hope. So that would be our affirmative team. They're saying the stem cell revolution is hype. Over here on the side of youth, and I'm averaging. Yeah, Tracy, you will sit with the old men. Over here, on the side of youth, we have that, when you're young, you are quite hopeful, aren't you? You're idealistic, naive, some might say. But we do have some heavyweight researchers over here as well. So here on our hope team, we've got this thing that, no, stem cell is not more hype than hope. It's certainly more hope than hype, or at least it's definitely not more hype than hope. I don't want to give away any debating strategies. Might have just let one slip there. But just a quick little get to know you, and then I'll introduce each of our speakers once they are due to take the stage for their very strict 5 minutes, after 4 minutes of which they will get a discreet yet effective warning klaxon sounding, and then the full school bell treatment at 5, okay? So there'll be no going over Martin. I've just been told to really emphasise that for you, that even with that American accent and charm, forget it. 5 minutes. In Tasmania, 5 minutes is 5 minutes, man. So, okay. So from the left, on our hype side, we've got Associate Professor Tracy Dixon. Let's give some love. We've got Professor Dom Rowe, he of the bow tie. We've got Professor Martin Pera. Team Hype. Here on the Hope side, we've got Dr. Joy. Now I have to make sure I don't mispronounce it because your partner is the Vice-Chancellor, so that would be bad. Dr. Joy Rathjen, yeah. Want to get that invite back next year, for sure. Dr. Kayleen Young. And Associate Professor Alex Hewitt. All right, we are just about ready to get underway here. So there's not too many rules, but I will be enforcing them because I'm the 9th child of 12 and you don't get a lot of special jobs in that role. So tonight my special jobs are being very hardline about timing and number of slides. So just forgive me when I just absolutely make you sit down and stop speaking at some stage, okay? It's nothing personal. It's just a little ego of a small child in a large family. Okay, so your job right now, because we're proper science here, we're going to kind of make a little bit of not a real study, but a sort of faux study here tonight. We're going to see if our teams are able to change your mind with the power of their persuasive arguments alone. Please don't distract the audience by standing up and pouring water while I'm talking. That kind of trickery, Associate Professor Dixon. Oh, right, yeah, I see what kind of a night we're going to have now. So what I want everyone to do is to place you've arrived here, you've heard nothing from the experts, your gut feeling on whether stem cell therapy or the stem cell revolution, God, and I have to take my glasses off to read. Okay, the stem cell revolution, if you think it's more hype than hope, I want you to tear off one of your hype sheets of paper. So you've all got 4 slips of paper, 2 for hope, 2 for hype. If you think it's more hype, tear off the hype. If you think it's not more hype, if you think it's hope, tear off the hope. We're going to gather all of those now. We'll be calling that baseline data. Okay, we're going to add up the hopes and hypes now beforehand. We'll announce that count a little later on. And after the debate, we'll do the whole shenanigans again and see if our extraordinary debaters have been able to change the average outcome. Let's not get down to tracking individuals. There seems to be altogether too much friendly chit-chat here between the two. Your adversaries, okay? So there's hype, there's hope. There's just enough of the, friendliness. Unless you're lulling each other into a false... Okay, gotcha, yeah. They're hardcore. Do we need any more time to gather the hope and hype slips? Okay, up the back there? Nearly there. I love the progress reports. We've got some hope, we've got some hype. We're going to be doing some serious counting. Probably get some... Is there a statistician in the house? We can do a proper breakdown. Results. Down here. Can't ring the bell or someone will stop speaking, yeah. Okay, does anyone have a slip that is yet to be placed in a ballot box? our work here is done. Okay, thanks everyone for that. Let's get started now with the actual debate. It's like a normal debate in that we will go affirmative, first negative, second negative, etc. The difference is there is no rebuttal. So everyone is simply giving a 5-minute presentation. There's no formal rebuttal. If you feel the urge to totally slag off or tear to shreds one of your opponent's arguments within the context of your own presentation, that's entirely up to you. But you will not have an allocated 1-minute period for rebuttal. So everyone has their 5 minutes that may or may not include legitimate sledging. And we're just going to go onesie, twosie, or however that works. And then at the end, we'll be asking for your vote again. So is everyone clear on the rules? That's enough from me, isn't it? I really did just need to take stage for a little bit longer. It's just that little child again. Our first speaker this evening deserves a warm and hearty welcome because he flew here at some ungodly hour from Melbourne. And I mean, being rescued from Melbourne itself, I think... Congratulations. And how glorious was today in Hobart. It couldn't have turned on a better, out view for Martin Perra. The only problem was he was locked in a dark room on a mysterious level of the building, engaged in teleconferences all day. So the poor sausage, see how he's all pale? No sunlight, hardly any coffee, and look at him sensibly drinking water, because he's never been to one of these debates before. Yeah. But no, Martin Perra is a very thoroughly experienced stem cell researcher. He's spent over 25 years researching human pluripotent stem cells. He was formerly at the University of Southern California and heading up the Stem Cell Research Centre there, and has been at the University of Melbourne since... I'd like a number, if that's okay. When did you go to Melbourne? About 5 years ago, and he's been in Hobart since about 5 a.m, okay? So we're delighted to have Martin here. He's going to take 5 minutes to present the case for hype, and he's going to do that with alacrity, he's going to do that with commitment, and he's going to do that probably without any rebuttal, I'm thinking, as the first speaker. So it's just my guess. Okay, Martin, are you ready to go? Okay, wait.
Bring my slides up. Don't start.
Okay, no, I'm not going to start. Once you've got your slides, your first of 3 slides, you tell me, and I'm going to press start for the magic 5 minutes. Ready?
Okay, go.
Go.
So having spent the past 20 years of my career as a strong advocate for this type of research, and in many public fora, this is a bit unusual for me, but it's instructive to take a real cold, hard look at where we've come in this period of time. And our motto this evening is nullius in verba. That's the motto of the Royal Society in the UK, and it means literally don't take anyone's word for it. Look hard at the evidence. Good advice 300 years ago, good advice today. So I think what I really want to focus in on is, we've done a lot of great research, we've learned a lot of really cool things, but what have we done for patients? So let's talk first about tissue stem cells. These are stem cells that are isolated from the tissues of the body. Well, blood stem cells, they're the standard of care for a number of cancers and blood disorders, well established, but that goes back 50 years or so ago. It's not the modern era, really, when these came into being. Limbal stem cells from the eye. These are approved now for use in corneal transplants. But again, the first clinical use dates back to the 1990s, so not too much new there. Now, there are literally thousands of trials with what we call mesenchymal stem cells. These are isolated from fat or bone marrow today. And thousands of trials going on, a lot of commercial activity. All of this is still experimental, and I'm going to bet that in most cases, the benefits from these therapies are going to be small and relatively short-lived. And then we can isolate stem cells from the brain, either the fetal or adult tissue. Again, these are in clinical trial. It's all experimental, and the biggest company in this sector, Stem Cells Inc., just went out of business a little while back because their big clinical trial failed. So it's not an easy road. Let's talk about pluripotent stem cells. These are stem cells derived from embryos or from reprogramming adult stem cells back to an embryonic state. And this looks pretty impressive. It looks like what we were promising 20 years ago. Age-related macular degeneration, common cause of blindness, Parkinson's disease, spinal cord injury, diabetes, myocardial infarction. All of these are going on, but they're very early stage. And all we've shown so far is that we aren't really doing anything horrible to the patient. There's really no evidence for benefit yet. So I think these early trials are unlikely to produce cures, and they may yield only modest or no benefit. I think that's the reality of it. I think we got to be prepared for adverse events. We don't understand how to look at the safety of this type of therapy. We don't understand what the long-term effects will be. We're really operating in the darkness here. And finally, these trials, frankly, are going to raise more scientific questions than they answer. It's great that they're going on, but it'll be a long time before these are standard of care for all of these conditions. So what is going on nowadays? Well, what is going on is really bad. What are patients being offered nowadays? And what they're being offered, and the map on the bottom right shows the distribution of this spread of unproven stem cell therapies around the world. And really, the only places you're safe from these charlatans are Africa and Greenland. Everywhere else, it's open season. These are treatments with no scientific basis whatsoever, but they're being offered to patients as hope for a whole range of diseases. And that's what this chart on the top left shows. It shows that stem cells are being offered now as cures for everything from Parkinson's disease to diabetes, to heart disease, to this whole broad, crazy range of conditions. No scientific evidence, no laboratory evidence, no clinical evidence whatsoever. And so this is the situation we're up against nowadays, and it's a very, very serious problem. And how has it arisen? It's arisen as a consequence of misunderstanding and misrepresentation of stem cell therapy. This is hype, not hope. And it's a very serious problem in the field. It's one we have particularly in Australia because our regulatory system has a loophole that's allowing this to go forward. And so I remain optimistic about this field. But at this stage, if we look hard at what we've done with the clinic, and what we've been able to do for patients, it's pretty limited, and we've got a really long road ahead. And that's from someone who's an advocate and who's been in this business a long time. Thank you.
Keep that applause going. Martin came in 20 seconds under the 5 minutes. Honestly. Here we go. 3, 2. That's your official end time, Martin. Well done, well done. That does not, however, mean, Joy, that you score an extra 20 seconds at any stage. I'm sorry about that. Now, interesting. A lot of therapies being offered. Very little evidence for them. I think that's spelled out. What was that word that you used? You're optimistic. Optimism. He did say charlatan bow tie, but he did also say he was optimistic. It does sound like a synonym for hope. But anyway, I'm not involved in the debate, so I would not be picking up on that point. All right, so that's our first speaker for hype. We're now moving over to the hope side of things. Next year, can we have two words that are a little bit different rather than hope and hype? I'm just not that skilled with the language. Okay, so our first speaker is Dr. Roy Rathjen. Now, while Martin has been working in this area for over 25 years, Joy's been working in the field of embryonic stem cells since 1991, which is why she's so much younger. She started off at the University of Adelaide with Peter Rathjen, so I guess we all know how that worked out. Not going there. Her current research focuses on learning how environmental cues and changing cell metabolism regulate the behaviour of embryonic stem cells and how they go in culture. So very much a stem cell scientist like Martin, and with a real interest in this area, and as we're about to hear, I'm sure, a conviction that there's a lot of hope in the stem cell revolution. Are you ready for your 5-minute count to begin?
I'm just going to check. I do animations. Am I not allowed to have animations?
You're allowed to have animations. I've been overruled by Alison. Apparently my petty grab for power has been revealed for what it is. All right, so enjoy your animations, see if I care. Okay, and your 5 minutes begins now.
Okay, thank you. So this debate is about hope. It's not really about what's happened in the past, it's what will happen in the future. Oh, yes. It's about the expectations of patients for stem cell therapy, and we can really talk about it is the hope of the population that stem cells will revolutionise medicine well-founded, or is it based on hype? Is it actually something we should be pursuing? And as Martin has very clearly demonstrated, there is a lot of hype around at the moment. The hype comes from an equation a bit like this. Martin called them charlatans, I call them snake oil salesmen. You give them the potential of a stem cell and you give them the hope of a patient, and what you get are failures, you get exploitation, and you also get, in some cases, dangerous practice. Some of your charlatans, or your snake oil salesmen, are really obvious. You can see them there. They come in spruiking a stem cell product with absolutely outrageous claims, like a cream that's actually fortified with a stem cell extract that will reduce your wrinkles. Most face creams do, but if you apply it to the breast, will also reduce breast cancer. Very unlikely. Some of your snake oil salesmen are much harder to pick. They are doctors with clinical rooms, or scientists with the authority of a white coat, who are trying to say that they can cure diseases with these untested treatments. And what they're doing is they're muddying the field and making it very hard for people to understand if there really is a reason to hope. So our job on the negative side, and it's strange being the positive voice in a negative side, which Kaylee and Alex and I are trying to tell you, is this is not the only game in town. There are a lot of places where stem cells really will and really are impacting clinical medicine at the moment. The first is that having stem cells in the current research in the world are making much better models of human disease, which leads to a better understanding of human disease, and there are new drugs and new approaches to therapy that are in the clinic now that are partly based on stem cell work. Alison has mentioned, and Martin mentioned, the fact that we have tissue-specific stem cells. All of you have stem cell populations in your body. And if we could recruit those stem cells, activate them and focus them down on treatment, then we would also have real treatments for therapy. And then finally, there is the therapy that you've heard of. Many diseases result from the fact that you have lost a population of cells, or a cell population has become dysfunctional. And these stem cells form other populations of cells, and so there's new opportunities for grafts and devices. So I wanted to finish by talking about one of these devices which I think shows enormous promise, to show where the field really is outside of the hype. This is a device being developed by a company called Viasight. It's called VCO1, and it's being developed for diabetes, a treatment for insulin-dependent diabetes. It's a bipartite device. You've got a membrane on the outside which encapsulates a population of cells. These cells are a human pancreatic population. The idea is that you'll put a little device in the side of the body, and it will be transplanted into the body. The membrane allows all the nutrients the cells need to stay happy and healthy in the body, and the membrane allows those cells to produce products that can then cross the membrane and act therapeutically. What the membrane also does is it stops any of the cells in the middle of the device no, it's not going to work, from escaping, and it stops any of the immune cells of the body from entering the device. What excites me about this device is this human pancreatic tissue. As you're probably all aware, human tissue is quite difficult to get hold of, but what you have is a beautiful human pancreas there. It's made up of the 3 cell types that we need, and they express insulin and the other proteins that we expect. Why am I excited about this particular device? All of the cells you can see here, all of the cells that will go into the device, are actually derived from a human embryonic stem cell. This is an unlimited source of human pancreatic cells that can be used in a therapy. This device has been used in diabetic mice, and it works beautifully. As you're sure, I'm also aware that we don't really have a huge need for treatments for diabetic mice. But this device has recently ended phase 1, phase 2 trials in the States, and people are being recruited and they're having these implants placed into their body. And we don't know if it'll work yet. It may not work in this formulation, but it possibly will work in later formulations. But it's when I see beautiful science like this and real possibilities for addressing a major disease of humankind that I still maintain a great deal of hope for stem cell science. And I think this rebuts the idea that it is all based on hype. Thank you.
Oh, thanks.
Sorry, Penny.
That's, oh, how's that for, did you hear that bell ringing? Did you hear that bell ringing? Look, I think that you've impressed a lot of people in the audience. Martin, obviously, completely unimpressed, just has left the scene of the crime. I think that was so he could get a better view of your amazing argument there, Joy. And I think now that we've heard a little from each side of the table, a little bit of hype, a little bit of hope, it's time to reveal where you sit. And I'm not sure if it's surprising or not. Who do you think came out ahead, hype or hope? Oh, they all sound so similar, who can tell? You're right. Hope is leading the field with 155 votes and hype 35 votes. So do we qualify that as audience bias? Okay, so clearly we've got a self-selected audience here. The issue is whether or not our teams can sway you. Can our hype team get that number up from the less than overwhelming 35 to something resembling, I don't know, 36? Maybe 37. Let's see how we go. But the arguments are coming thick and fast. They're powerful and they're convincing. Will they do the trick for you? He's keen to get going. I know he's very much committed to his 5-minute time slot. He's going to have to wait for me to have my moment in the light. Professor Marrow is Foundation Professor of Neurology in the Faculty of Medicine and Health Science at Macquarie University in Sydney. He's from Sydney, but be nice, okay? He's come all this way. He's the chair of Macquarie Neurology. So he's a consultant neurologist and he's a researcher. In fact, he's the co-founder of the Australian Motor Neurone Disease Registry. And his two research and clinical interests are in Parkinson's disease. His PhD was in working out the way that disease developed in people, and his other areas, that's his main area of practice, his other areas, motor neurone disease. So he's obviously very much interested in these areas for which so much promise has been touted by stem cell therapies or by the proponents. So, and yet, this evening, he's arguing for the side of hype and, as I've pointed out a number of times, embodying the snake oil role with that very fetching bow tie. Dom has also warned early on, he's a self-proclaimed berserker. I did not know what that was. A berserker is a term from the Middle Ages, which is the guys that they would send in when they really wanted to win the war or the battle, because they were the nut jobs who had no off switch, okay? They just kept, gah, 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 kill, kill. He's like a human Dalek. Okay, so he's pretty committed tonight to winning the argument. He will not be turned off except by the power of the 5-minute bell ringing, which he'll be very respectful of. Join me in welcoming Professor Dominic Rowe. Are you ready, Dom, for your 5 minutes?
Thanks, Bernie. So I'm going to bring you back to reality from cartoons of Donald Trump. Today, 2 people died from motor neurone disease in Australia. 800 Australians die each year from this horrible disease. These people fall victim to the hope argument in motor neuron disease. Coming again to a Latin phrase, and we had one from Martin, but primum non nocere is the cornerstone of medicine, and that means, first of all, do no harm. So the stem cell therapy in motor neuron disease, and I will restrict myself to neurological diseases, is all hype, and you will get robbed. There's a cartoon for you, Joy. I'm going to give you three cases from my practice. I could have given you many more, of people who have died from motor neurone disease who have pursued this snake oil. So the first is a 52-year-old female medical practitioner who started in April 2008 with weakness in her left leg. Her weakness progressed. She stopped work. She went to Lourdes thinking that that was going to help. She actually applied to the autologous stem cell team at St Vincent's Hospital that do all sorts of autologous stem cell from bone marrow for various different indications, some of which are quite fringe, and they turned her down on the available evidence. She had a trial of anti-parasitic therapy. She then applied to a company called Regenius in Sydney. Regenius are ASX listed. They are in Pymble, and they produce mesenchymal stem cells from people's fat. She underwent intravenous mesenchymal stem cell transplantation in a private hospital in Sydney. She continued to progress, unable to stand, progressive spasticity. We sometimes, when people can't swallow, we put a tube into their tummy called a percutaneous endoscopic gastrostomy. She declined any ventilatory support. She died at home, 40 months from diagnosis, 48 months from symptom onset. Mesenchymal stem cells, all hype. The next patient, a 60-year-old female union delegate. And I'm sorry to be so grim about this, but this is reality. This is what I deal with in the clinic each day, not rubbish in a science lab. This lady presented in 2012. She fell, fractured her left ankle. She eventually was diagnosed with motor neurone disease. She came to see me in the middle of 2014. Then she was already a resident in a nursing home with spastic tetraparesis. She could not stand, she couldn't transfer. She was undergoing mesenchymal stem cell therapy by a well-known charlatan in Sydney called Ralph Bright, who was charging her $4,400 a month plus peptides for which there is no scientific basis per month to treat her motor neurone disease. Surprise, surprise, they didn't help her. She rejected the routine therapy. She underwent an emergency percutaneous gastrostomy because she could no longer swallow. Even after putting in a peg, she was so deluded that this therapy was going to help her, she kept on going. The total cost of her therapy is estimated to have been $250,000, and she died. The last patient, and I'll speed through this, 75-year-old male grazier, ended up with thoracic motor neurone disease. He went to Bonn. For 1 day's treatment in Bonn cost him €14,000, where they took his own bone marrow, injected it into his muscle, blood, and into his spinal fluid. Give me a break. Cloud cuckoo land. Nuts. He progressed. He ended up in a nursing home and he died. At Macquarie, we actually look after people and they generously donate their brains and spinal cords so that when it's got feathers and webbed feet and goes quack, 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 it's a duck. So we actually confirmed that John died from motor neurone disease. His stem cell therapy did not help him one iota. So it's all hype, rubbish.
Thanks very much for that, Dom. Tracy, how are you feeling with your? How was it described, that play school science? What was it? That rubbish benchtop laboratory stuff you do for a living? Yeah, no good. Team love, team love. Oh, it'll all be settled in the car park later, I'm sure. Well, that was very sobering and very much a hard hit, I think, from Team Hype there. What is Team Hope going to come back with? Well, I think we know we're in good hands, because not only is Dr. Kayleen Young our next speaker for Team Hope, not only has she had some remarkable breakthroughs in her, I mean, look at her, she's 12. And she's had an exceptional research career to date, was part of a team that discovered a kind of brain stem cell that actually repairs the insulation material around nerve cells. I mean, that's the problem in MS. This is serious stuff. This is a stem cell that can do that sort of work, obviously in the laboratory, in the playland of the laboratory. But we'll be hearing, so, you know, we're with a serious, like with our other speakers, a serious stem cell researcher here, but forget about that. I have it on good authority that Kayleen Young was the best debater at her school. Oh, yeah. I'm thinking year 12, Champions League, affirmative, negative, mattered not. Kayleen Young wiped the floor with the opposition. And that is what we want to see right now. Fighting form. Kayleen, have you got it here tonight? Well, she did have a champagne before we kicked off, so I reckon she's gunning for it. She's ready. Do you want this mic or the other one, Kayleen? You'll have this one. I'll give you a moment to... I'll give you a moment. I'll give you a moment so I can get my timer sorted. Are you ready to proceed, Kayleen? She's got her serious face on now. Yeah. This looks fun. Okay. Kayleen, your time starts now.
Okay, I'm not 14 because I'm about to tell you that I've grown up in an incredible era of stem cell research that has actually seen stem cells become an integral part of modern medicine. And the interest in stem cells was even around in the 70s, and it was already quite controversial, with the media coining the term test tube babies for children that were born as a result of IVF. Now, it says here a lot of love and a little science equals one sweet baby. But actually, it was a lot of stem cell research that resulted from Professor Robert Edwards' laboratory with him and his colleagues that resulted in the birth of Louise Brown in 1978. Now, a lot of you probably don't think of IVF as being a stem cell procedure, but actually, if you take one egg and one sperm, what you get is the ultimate stem cell that gives rise to all of the cells in the body. And so from this point of view, IVF is actually an excellent example of stem cell science that took a long time, many decades in the development, and received considerable media attention, but has actually gone on and progressed to become what is a relatively common fertility therapy that has helped many Australians realise the hope of starting a family. So if we jump forward now to the 80s, this is a decade where we actually see about 40 years of research into blood stem cells really come together to result in a treatment for immunodeficiency disorders as well as certain types of cancers. So I'll show you here a movie. This is... This is a white blood cell attacking and killing off a foreign invading cell. This is another white blood cell engulfing and killing a yeast cell. Now, this is exactly what what white blood cells do, excluding the burping. But they only live, they defend our bodies against infection, but they only live for a short amount of time. So what we need is stem cells to constantly be making new white blood cells. And these stem cells reside here in the bone marrow, in the middle of our bones. Now, I'm sure many of you have heard of bone marrow transplants. They've been developed and refined all the way through sort of from the 50s through to the 80s. And this is actually a stem cell transplant therapy, so that you take these cells from a donor, they get put into a patient, the recipient then has these new blood stem cells that are in their own bone marrow that will make lots of new white blood cells that will protect them against disease and fight against their immune conditions. But the 80s also saw an important discovery from an Australian scientist, Professor Don Metcalf. He discovered that you could have a hormone injection that will then trigger our body's own blood stem cells to release a whole bunch of new white blood cells into the circulation to fight disease. Now, this was important because at the time it was discovered that chemotherapy was actually causing cancer patients to have a really big drop in their white blood cells so that they were unable to fight off infection. And so this therapy, which relies on stem cells, our own stem cells, has actually affected 20 million patients since its implementation in the 80s and allowed them to undergo more rigorous chemotherapy treatments in the legitimate hope that they will actually then be able to overcome their cancer. So this brings us then to the 90s and the discovery of brain stem cells. And if I show you a cell here, the one up in the side is actually currently dividing, and the one on this side has just finished undergoing a cell division. Now, we know that brain stem cells make new cells in the brain every day. They're important for mood and memory. We've got a lot of challenges to overcome, I'm not denying that. At the moment, the first stem cell therapy that actually triggers our own brain stem cells to try and repair the nervous system is currently in trial in multiple sclerosis patients. And so while we have a lot of challenges, I suggest that as modern stem cell researchers, we don't run away from those problems, like I think these cells over here are running away from this scary one over this side. But we instead sort of embrace those challenges and say, okay, what we have at the moment is something that is still looking incredibly promising and hopeful for future brain repair.
She's so close to coming in right on the 5 minutes that I'm almost tempted to ignore the fact that there were more than 3 slides in that presentation. But, you know, she glamoured me with 70s, 80s and 90s. I was so busy reliving Dynasty's great era and Joan's makeup and Linda's shoulder pads that I barely noticed that they were extraneous to the number of slides permitted. Let's put it together again. Now, what did I say? That's Debating Strategy 101. Redefine the topic to something that suits you. Give examples of stem cell therapies that everyone's familiar with that have been around for ages. Nice work. Well done, Kayleen. Well played. I'm sure that did not go unnoticed by Associate Professor Tracy Dixon, who's probably about to have a little go at... Yeah, no, I thought that might be what's coming, yeah. I mean, I just... Like, you can see the professionalism at work here. It's terrific. Now, Tracy is Deputy Director of the Menzies Institute for Medical Research, and she's the lead of the Institute's Neurodegenerative Disease and Brain Injury Research team. So while Kayleen is the team leader over on this side, Tracy calls the shots over on Team Hype, all right? So as well as all of that very official stuff, I have been told that by also a very reliable source that the Dixon Lab, Tracy's research group, they're known as the Fun Lab. Okay, so the expectations for this particular argument have just gone through the roof, all right? So no pressure, Tracy, but, you know, give us your best shot. Join me in welcoming... Oops. And are you just about ready? I will hand you the microphone as soon as... Oh, because she's so low. You ready to go?
Yeah, I'm ready.
Okay, good.
So I'm not afraid of debating team experts. I'm not afraid at all. I'm going to bring my A game. So we've heard already about mechanism, understanding, blah, blah, blah. We're all interested in cures, and it's the promise of cures that has fueled this debate for many, many years. So tonight I'm going to tell you something about that we all agree on. We've all been to high school, we've got PhDs, we've all been to university, but I'm going to save you all of that. I'm going to save you the books, I'm going to save you the lectures, I'm going to save you the university fees, and that's our Vice-Chancellor. What everybody on both teams agrees is this: the brain is complicated. And we can't just fix it with putting in some cells and crossing our fingers, and I'll tell you why. The brain is complicated. Neurons, they're not your average cell. They're not that blue blob at the top. They're that beautiful cell down the bottom. They're incredibly specialised. They have many different compartments. And the cartoon there says, you want to be a brain cell, you've got to study. You goof off, you'll end up in the rectum. So be careful. Not all neurons are the same. There's billions of neurons in our brains and there's 200 different types. Why would we think it's as simple as just making a neuron in a lab and shooting it into your brain? They're not all the same. We need to make connections. Let's see if I can whiz this. There are millions of miles of connections in our brain, and the picture at the top, every different colour there shows you a different axon tract happening in our brain, and down the bottom you see just one sensory axon making its path, centimetres it has to grow in a mouse, up to a metre in a person to make the connection to the correct target. It's difficult. We need to make the right connections. Every single neuron has about 7,000 synapses, 7,000 different connections in the brain, and they can't just talk to anyone. Down the bottom you see a picture, one little red cell, hopefully you can see it, but in green, all the cells throughout the brain that it's connected to. They're not just the cells nearby that they hold hands with, they're connected to cells that are distances away. It's complicated. So what are the chances? I'm going to ask our 3 opposition members to be part of this experiment. Please stand up, my friends. So imagine that Alex and Kayleen and Joy, they're stem cells. They've been turned into neurons. They've been injected into the brain. They're going to cure Alzheimer's disease. But hang on, Alex, you're not even a neuron. Somebody made a mistake in the lab. You're a rectum cell. Sit down, my friend. You're not viable. Joy, you're a neuron, but you're not the right type. You're in the wrong place, my friend. Please sit down. Kayleen, you're the only one who's actually a viable neuron, but you've got to make a connection. Now, who, my friend, do you want to connect with? Just one person in the crowd here is the appropriate connection for Kayleen. So have a go. Have a guess. The guy in the orange top. It's a miracle. Everybody have a look at your chair, and if you've got a red ribbon, wave your hand. Wrapped around the handle. If you've got a red ribbon, wave your hand. This gentleman. So this gentleman is the perfect match for Kayleen. She couldn't do it. She couldn't make the right connection when there was only 200 people here. What if there were millions of people here? What if she had to make 7,000 appropriate connections out of those million people? It is way too hard. There is no hope. So I think we've shown tonight that we have the numbers to prove it, we have the stories to prove it, and we have the science to prove it, that stem cell therapy is all hype, no hope.
How are you feeling after that little diagnosis?
The rectum.
The rectum, yeah. She's messing with your head, yeah. 7,000, is that true? I like how you're sticking with the facts tonight, that's good. I had no idea that every neuron in the brain, on average, 7,000 connections. Well, I mean, we could all say that, but in your case, it seems to be true. Fantastic presentation. Look, I think, don't forget, to be declared the winners, they only have to nudge up from 35 votes. Was it 35? Is that correct? 35 to 36 votes. I don't know. Was that argument then enough to do it in combination, obviously, with the rest of her team, or will Alex with that advantage of being the last speaker, be able to reinforce the absolute bias that exists in this room towards the hope side of things. Alex is not just a stem cell researcher, he's a clinician as well. So he's an ophthalmologist. When I pointed out to Dom that we had another clinician and that he was an ophthalmologist, he said, "Oh, they're not real doctors." So just if you need a little fire in the belly, I don't know if that's going to help. I just thought I'd mention it. So not only is he a researcher, let me just read this properly. He recently received a Research Excellence Award as the top-ranked NHMRC Practitioner Fellowship applicant in Australia for his work on patient-specific stem lines. I had to ask Alex, what does that mean? He said, it means there's no more money for research. Okay, so yeah, that's pretty much, I think, a fair summation of what the outcome was. But look, this is it. Alex, you're the man. You've got the final say in this debate. Are you ready for your 5 minutes to begin? Then how about I get the clock? Okay, over to you, Dr. Professor, Associate Professor Alex Hewitt. Woo.
So I guess I certainly want to primarily focus on the 35 of you who don't believe in the hope from this technology and from this work, and I certainly don't want the other 116 of you there who voted in the hope to believe that I'm a rectum or that my profession is totally nonsensical. I do want you all to leave here tonight with a realistic sense of optimism. The stem cell bucket is over half full. As Kayleen and Martin outlined, their stem cells from blood is already being used to cure leukemia. And as Joy described, stem cell-based therapies for diabetes is bloody close. But we do acknowledge that there are undue and unwarranted hype for quack stem cell therapies. But as an informed audience, it's important that you leave here tonight with a healthy sense of scepticism about the stem cell hype. You don't need to pay $10,000 to go off to Sydney or to get some fat sucked out of your tummy, jizzied up and injected back into your joints to get a cure for your arthritis. You don't need to travel to India or China or Russia or even America to get a stem cell therapy that has no proof. Is it not absolutely amazing that we can now take an adult cell, such as a skin cell or a hair cell, and turn that back into a stem cell, and then from that stem cell differentiate that to different cell types like heart cells or eye cells or even brain cells? Using that technology, we can study the cells at the back of your eye without actually taking a sample from the back of your eye. It's early days, the work is still going, but we're inching closer. It's possible now for us to study disease cells from you that will provide further insight into disease mechanisms that will hopefully uncover new therapeutic targets. The tools for biological discovery have never been sharper, but advances do take time. To my mind, one of the best examples of progress over time actually comes from the Olympics, and it must blow Larissa Latino's mind seeing the current vault routines used in the Olympics. You can see her winning performance in 1956, and that required to achieve gold 56 years later. It's not even been 20 years since Martin Perra discovered how to keep a human embryonic stem cell alive in the laboratory, and this year marks 10 years. It marks the 10-year anniversary since the Nobel Prize-winning discovery of how to turn an adult cell back into a stem cell. Can you imagine the sort of backflips that we'll be able to make stem cells do in another 20 years? On the scientific front, most of the major breakthroughs take generally 50 to 60 years. It took over 60 years to go from the Wright brothers to putting man on the moon. It took 70 years to advance from the first electric general-purpose computer to a Google AI program that was capable of beating a grandmaster at Go, the most complex game ever developed. And 60 years after deciphering the structure of DNA, we can now edit it with high fidelity. Merging these gene editing techniques with stem cell technology will expedite progress. Adapting specific proteins used by bacteria to fight off viruses to now work in mammalian cells means that we can now, with very high accuracy and fidelity, go in and correct specific disease-causing variants in stem cells. It is possible with time that we could then use these corrected stem cells to be reimplanted back into your body. Finally, the major reason why I feel that you need to be truly hopeful about stem cells is the fact that there is a small army of amazingly skilled scientists and clinicians working very hard to develop better tests and treatments for all diseases. Ongoing research with stem cells will mean that your grandchildren will have a dramatically different disease trajectory to you. The changes that will come in the next few years will be breathtaking. So vote for hope.
Vote for hope. There it is. That's the words that we end tonight's plea from the debate teams with. Let's not overlook some of the other words we've heard. One of my favourites comes from Alex, from the award-winning applicant, research applicant, explaining how the stem cells that come from your fat tissue are jizzied up before they're put back in your body. I mean, as a science communicator, I'm almost brought to tears with the... like, to be able to get that across so simply. It was art. It wasn't just science, it was art and beauty. I think we've ended on a very, very strong note there. I think it's very tough. The competition's very tough, very firm, but you've heard the words from both sides tonight. You've got your special bits of paper. It's down to you now. Are you going to vote hope or hype after hearing these very balanced, very rigorous... No, we gave ample, but nice touch, Joy. So we are now collecting your votes. Just remember, you're voting on what you've heard from the debaters, from these experts in jizzying and other forms of science, from practitioners, from highly skilled people with many bits of book learning and the occasional rectum. You've heard it all here tonight. Your mind may have been swayed, it may not. In which direction? We won't know until the last ballot is counted. I feel like I'm in church, getting a bit ethereal. How are we going with the counts there? Do we have more votes to be collected? Oh. Do we have a prize for Mr. Red Ribbon guy? I feel like, just the ribbon itself, that's reward enough, yeah. And what about, it's what, yeah, you're going to not have lunch with Kayleen because she just couldn't make it work. What about orange shirt guy? He's totally disappeared. Oh no, the young fella. And one thing I think we have to take heart in, once we were finally able to, I love the GIF. See, the young people use GIFs like that. They're, the Olympic thing, I think you made your argument very well apart. It wasn't actually a backflip that she was doing, it was a forward. But anyway, apart from that, I like how, what was the, I have a point. Maybe I don't. What were we talking about? Oh yeah, the 50-year thing. So 50 years on average, Martin's work 25 years ago, so we're thinking within 25 years, which before you retire, more importantly, I'll only be 76 and a half. So I think there's some hope that there'll be decent therapies before I'm completely done and dusted. So please, people, get back to work, would you? Do we have any more slips to be collected? Okay, no. So now you have got about 15 minutes free-ranging time. Here's a microphone for our respondents. I am coming to you. Put your hand up if you've got a question, and let's hit our experts with any questions relevant to stem cell therapies, to the kind of research, the hype, the hope. Thanks for being the first question right at the opposite end of the theatre. And just quickly your name and your question, and if it's for one person in particular.
Hi, my name's Kai. My question is just to everyone, and it's just a curiosity, really. Are epigenetic changes carried forward to adult pluripotent stem cells, and how are telomere length affected as well by the creation of adult pluripotent stem cells?
Right, so just a simple question. First off, let's just, see them hand the microphone to other people. Yours. You know yours is not a call. Have you not played sport? Okay, Martin, yours.
So I think I understand the question. What this question is about is transforming a cell from the adult body back to the pluripotent state. And the question is that done with fidelity? I think that really what it relates to, because we don't change the genes. What we change is the way the genes are read out. This is called epigenetics. And are we really making a proper replica of an embryonic cell, or are we making something that's not quite right, that'll go off the rails, etc.? And are we restoring the capacity for that cell to grow indefinitely the way embryonic cells do? And those are really important questions. They've been in the field since the methodology was first described. I think as we go on, the consensus more and more in the field is that really these induced pluripotent stem cells, these cells reprogrammed from adult tissue, are really pretty close to embryonic cells. And what we used to think was differences between those cells and embryonic cells are now interestingly put down to patient-specific genetic differences. In other words, it's the genome of the patient that's accounting for a fascinating area. But I'd say more and more we're thinking these cells are bioequivalent.
Okay, thanks so much, Martin. That was a great answer. And we'll come back, if we don't have any other questions, we'll come back to that second question about telomeres. But here's our second question.
Question to the affirmative. My name's Sean, by the way. If you believe that what you're doing is a waste of time, why are you doing it?
I look after people with diseases. I try and convince them not to follow stem cells for neurological diseases. I don't think that's a waste of time. By looking after people well and trying to understand the biology of the disease, we will make much more change in the progression of the disease than trying to injure an organ that's so complicated, like the brain, that's been ravaged by a disease like motor neuron disease for 5 or 10 years, or Parkinson's disease that's been ravaged for 15 to 20 years, and trying to fix that. Last year in North America, industry spent $4 billion conservatively on cellular therapies to repair the nervous system. If they spent that same money on trying to understand the diseases in the first place, we'd be a lot further ahead.
Thanks, Tom. And our next question.
In the case of a stroke victim that's had a bleed in the brain, what would be the physical mechanism for stem cells to solve that problem?
Yours.
I'm not sure at this stage we have the answer to that question. I think the affirmative side have made it very clear that the brain is very complex. There's a lot of other places that stem cell therapies will work. But one of the ways I think people are looking at how you might use stem cells in these sorts of treatments is in the recruitment of endogenous stem cells to try and improve the repair of the organ in the way that endogenous cells do, and also to try and reduce the scarring. It's not my area of expertise though, so if anyone on the affirmative wants to come in.
Gee, they're really leaping at that, aren't they? It's
I can talk under cement, Bernie. Look, cellular therapy for people who've had an injury to the brain, either an ischemic stroke where the artery is blocked off and the brain dies, or if you have a hemorrhage and destroys the brain by pressure. There are no proven therapies in humans with cellular techniques to actually improve outcome at all in 2016. All hype.
They've already voted, okay? You can lay off the hype line.
I told you what I was.
Yeah.
I'm not going to
Berserker.
stop until there's a carcass on the floor.
Yeah. Okay.
I actually agree with them. There isn't any cellular therapies at the moment. What we're looking at is the potential for therapies in the future.
I think you call that hope.
That's what I call hope,
Yeah.
yes.
And I think, okay. You know, Ben, it's my job to wield the microphone. I'm not loving how you just come around and give... Anyway, yes, please, ask your question.
Hi, my name is Bronwyn. I'm only one of 6 and I'm the last in the group, so I know
I share your
exactly
pain.
what it's like, yes. My question is, what do you as scientists do to combat the hype that is out there? And where does it come, where does the hype stem from, if you're... go for the pun, sorry.
That is a really important and more interesting question than what we're debating tonight. So from the beginning of this field, it was very difficult to strike a balance in communicating to the public the potential for this research, whilst at the same time trying to get across how difficult and complicated you've heard. And it's a fine line that we have had to walk from the beginning, because in the beginning, you may not remember, there was an awful lot of opposition to this research for all sorts of reasons. And we felt, we still feel, we have very good reasons for doing it. I think one of the biggest barriers is really around scientific literacy, and that's true not only in our field, but in many, many fields. Every day, nations take huge decisions around very, very complicated issues involving science and technology that we don't really understand. And clearly, the understanding of the general public, these things are hard enough for us to work at, but the understanding of the general public of this field in general is pretty rudimentary. I had a colleague who once testified in front of the United States Senate when this field was first taking off. He said he was told to assume a 9th grade education at the United States Senate. So I think this whole issue of scientific literacy is a hugely important one, and hopefully events like this contribute to, helping that along.
Fantastic. Great question. Oh, here we go. We've got a run
Can
of
I just
questions.
add something in, Bernie? Can I add something in?
Yep.
So, a lot of the problem is at the regulatory level. Okay?
Sorry?
A lot of the problem is at the regulatory level. So I mentioned 2 companies that work inside the TGA to exploit people's hope that they're going to fix their lethal disease. They're in a corner. They're going to die. They have the worst disease that you can imagine. It robs you of your mobility, your speech, your breathing, everything. And you can understand the motivation of these people to pursue any therapy that might be possible. And it comes with a shiny card and a website. The TGA has not cleared up this area. So you can take someone's fat, in me there's probably a little bit too much, and you can inject it anywhere legally in Australia now and charge people, and I'm not allowed to swear, am I? No. You can charge people a lot of money and get away with it. And that's why... Sorry?
If it's their own fat.
If it's their own fat. And they don't manipulate it, and they call it mesenchymal stem cells, and they produce beautiful charts and graphs and everything. And the second case that I showed you, she was charged over $250,000, and it didn't help her a cent. And don't get me wrong, I think stem cell biology is fascinating. It's an important area of science, and it tells us a lot of things about what turns a cell into a cell. But in terms of cellular therapies for neurological disease, it's not there.
Great, excellent points. Thanks, everyone.
I was just going to add the other thing about the people who are doing this inside the TGA. Unlike what I showed you from Viaside and the people who are trying to get therapies through, they don't have to show they work. There isn't anything that says that they work. And they use the promise, they use our reputations
to feed people's hopes. And as Dom says, when people are dying, or even their quality of life is affected, they will do anything. And so they are very, very vulnerable. And so there's a lot of work being done by a lot of people to try and educate people on the vulnerability. There's some very good material on the International Society of Stem Cell Researchers website, and also on the Stem Cells Australia website that Martin heads up, Stem Cells Australia. If you want to follow this up more, it's worth doing, because it is important to understand these sites look really glossy. I remember looking at one site that had been set up by a company in Malaysia. They were German scientists, and they come with all that very German properness. And they were promising therapies for all sorts of diseases. And when you actually looked into it, what they were injecting into patients wasn't stem cells from their own fat. They had a very, very special population of rabbits, okay? And they were taking stem cells from the rabbits, and they were promising to cure diseases like Alzheimer's and autism. And it's really frightening. So it's really important to be aware. Sorry.
No, not at all. I think that was a really important question and beautifully answered. So thanks, everyone. The hype comes from green.
Or it comes from scientists who honestly believe that we're just going to circumvent this trial business because, of course, it's going to work.
Yeah, a testimonial.
But some of it's...
Yeah, testimonials on shiny websites are not actual evidence. So, yeah, your question.
There are different types of stem cells, and there are also different types of cells in which they could differentiate to. Which kinds of conditions do you think there is most hype, and which conditions do you think there is just most hope as well?
Interesting. Which conditions most hype and which conditions most hope? The hand pass.
Yeah.
No.
I think our case, and that everybody on the panel agrees, that it's the neurological conditions that are often sold with the most hope and probably are the most hype, because it's so complicated and it requires a very different type of cell. But there is hope for stem cell therapies, as we heard about, making different growth factors, putting back into the body, cells that play important roles. Hormones, for example, those are cells that you can imagine a therapy being evolved for. But the neurological ones, it's really hard to see.
Fantastic. Last question. Do we still have that? Yep.
Hi, I'm Chantel. I've got 3 children, and I saved my umbilical cord blood, and I have their stem cells stored. And my oldest is 13 now, and I'm interested, any of you could answer this, I'm sure. Do you think they will ever be useful to them during their lives?
How much does that cost you per year to store them?
It was $150 a year, and I've paid for them all till they're 18 now. My oldest is 13 now, and it cost me, trying to remember back, about $1,000 a child. This was in New Zealand when the cord bank was new, and my first child was number 55, and my third child, by the time his was stored, was 2,364. That was his number. So it grew, like 13 years ago, not many people were doing it. The lab was really new, and he was, the 55th kid. I think I'm the only person, the first person that had 3 children to have the cord bank blood saved. And I just wonder whether it was a worthwhile exercise in your opinion, and whether it will ever come to any fruition for them.
Fantastic, thank you.
I'll be brief. There were 26 odd million people in Australia. Only 400 children per year develop some form of haematologic malignancy like leukemia. So in that instance, the cord blood may well be a very helpful therapy for, in that very rare event that a child develops a haematologic malignancy. For every other reason, there isn't really an indication, to my understanding, unless someone can shed light.
Anyone want to?
So it's a low probability event, but
it's just like buying insurance, and in time I'll sleep well at night, don't worry about it.
They've already voted, Rectum.
But again, like insurance, it preys on your fear of something that isn't very quantifiable, but this is quantifiable. 400 kids per year in Australia may develop some form of haematologic malignancy. Each one is a tragedy. Having said that, therapies now in haematologic malignancy, with your first course of chemo, cure rates are up to 70 to 80% in forms of chemo.
And it'd be sensible for Australia and or New Zealand to have an Australia-wide banking service that does it. Would be a smarter, so you could potentially HLA match people and all that sort of stuff. That's where I would invest.
Okay, that is it for our Q&A time. It is now time to reveal the results of the vote. Okay, so Australia, or this room full of Australians, you have spoken, and let me tell you that Hope has come in with a total of 129, meaning hype has gone through the roof and almost doubled to 55. Woo! So, even though the majority of people still favour the Hope argument, clearly the debate tonight has been won, I think we've got to agree, has been won by Team Hype, who are very keen to stand up and take their applause. Tracy's, yes! And I think that speaks very well to the concern and issue of making sure that people are a bit more informed about these, about this issue that I think everyone really shares the same concerns about, even if there was some fairly legitimate and some illegitimate sledging going on tonight. Would you join me in thanking, once again, our spectacular debaters who've done a terrific job? Tracy, Dom, Martin, Joy, Kayleen and Alex. Hype, Hope, they've nailed it. You've had your say. It's been an absolute pleasure, and congratulations on taking part in such a terrific event. And I'll just hand you back to Alison. Thank you.
Thank you, Bernie. It just gives me the job of wrapping things up and thanking our debaters very much. Thank you very much to Bernie. Great having you here with our debate, and I hope you can come again when we do this next year. Really, to all of you who've attended, you've been a great audience, great participators. We really enjoy putting this event on at Menzies. We do a number of community events. This one's certainly my favourite. If this is the first Menzies event you've come to, we really hope to see you again. Our website is a really good, is the place to go to check out what we have that's coming up. So thanks to all of you. We have just some small gifts to give our panellists. It includes some stem ginger. That was the nearest we could do on a theme. Yeah, okay. Slightly different ones for our interstate guests, I think. Thank you. Thank you. And one for Bernie. Oh, that's for the others. Okay. And last but not least, a small one for Bernie.
Oh, thank you very much.
Yeah. And just finally, obviously it takes quite a lot of work to put an event like today on, not just the work of the teams in preparing, but also from Miranda Harmon and her team who've organised all the logistics and the media and everything else. It's been a big effort. So thank you, Miranda, and all her team. So that's it. Thank you very much, and look forward to seeing you again another time, and especially for the debate next year. Thank you.